Hello, welcome to an overview of the polymerase chain reaction testing. The COVID-19 RT-PCR test is a real-time reverse transcription polymerase chain test that detects nucleic acid from SARS-CoV-2 RNA. The PCR test amplifies virus's genetic material. If the virus is detected in the body, it becomes amplified, allowing even the smallest amount of virus DNA to become detectable. In the figure below, a quick overview of the procedure is shown. Original DNA is denatured. A DNA primer then attaches itself to the complementary sequence of the single-stranded RNA. Taq polymerase can then build two new strands of DNA containing one old and one new strand. The cycle continues 30 to 40 times, which can lead to 1 billion exact copies of original DNA segments. As stated, PCR is a nucleic acid test that works by amplifying small amounts of genetic material. The thermocycler is used for the heating and cooling step. The sample is combined with probes, primers, DNA polymerase, reverse transcriptase, buffer, and DNTPs, which are artificial nucleotides that are used for the synthesis of DNA. And the combination goes through the first annealing phase, which is first complementary DNA that is formed. The probes contain Q and R dyes to anneal to the sections of the targeted genetic sequence. The polymerase extends the primers out of the probe, and this releases R generating fluorescence. This is a picture of the thermocycler. Um, the thermocycle will continuously repeat itself in order to amplify the genetic material with every cycle it goes through. If a certain level of fluorescence is obtained through the cycle threshold, we know a positive test was yielded. Basically, the chemicals bring about a fluorescence light if SARS-CoV-2 is active in the sample. The RT-PCR test is utilizing fluorescence resonance energy transfer during this. So here we have a diagram showing the process a little better. The DNA denatures at around 94 to 96 degrees Celsius, and it becomes single-stranded DNA, and then it is cooled so that the probes and the primers can attach. And this, as the cDNA is formed, the primer is released and fluorescence is activated at a slightly higher temperature, so 72 degrees Celsius. And the probe is released, the fluorescence is activated and recorded by the thermocycler. This process can be repeated continuously, measuring the fluorescence and flagging the presence of the virus. This test can be 100% accurate, but it could take quite a bit of time, and it requires specialized equipment. Here we have a figure of the primer binding to the RNA, single-stranded RNA, reverse transcriptase, and the DNTPs are present as well. The reverse transcriptase comes in and builds the complementary DNA with the DNTPs, and you're left with complementary DNA and the amplification of it with the specific primers and the Taq polymerase. There's two main methods to do to obtain your sample for the PCR test. One is a nasal swab, which is a little discomforting, and it can also put you close to the healthcare workers, which could increase the spread of the virus. The saliva PCR test is more non-invasive, and you can get a result in 24 to 48 hours. It was recently developed in New Jersey. The RT-PCR test is favored because of its amplification of the virus and sensitivity, with even the smallest amount of the virus present in the sample, as we saw in the earlier slide. So there are several different testing methods, but the PCR is a viral test and molecular test for nucleic acid. Amplification is usually done through a nasal or throat swab or saliva. It can be the same day at some locations, depending on I think staff availability and how many requests are taken. Because we saw that it takes 3 to 4 hours uh, to get the result and the amplification of the DNA. It's very accurate and it's a gold standard for COVID-19. It diagnoses you with coronavirus infection essentially, and it'll show if you ever were infected or in the past as well. Research has been coming a long way for COVID-19. In China, CT scans were used to compare the lungs of the patients and healthy ones before the PCR. The use of PCR and other methods of testing, RT-PCR testing is important as it allows for the fast detection of the disease. Prolonged detection can lead to further spread and longevity of the disease, so this is good to catch the disease uh, and locate the spread of it. And PCR testing is necessary for fast detection, isolation, and to overall help the eradication of the COVID-19.
Hello, I will be discussing about some of the research that has been done on the human coronaviruses as well as the new variant for COVID-19. The first research that has been done is on a research paper titled A Melting Curve-Based Multiplex RT-qPCR Assay for the Simultaneous Detection of 4 Human Coronaviruses. The 4 variants for this are just going to be 229E, OC43, NL63, and HKU1. Next to the listed coronavirus variants is going to be the primer set, their name, and then the sequencing as well. The melting curves for the 4 human coronavirus variants were determined using the first set of the primers. And then these melting curves can be shown in figure 8 right over here. And as you see, there is an overlap between the 229E and the OC43 variant. And in order to make a clear distinguishment, the primer for the OC43 variant was modified with a GC-rich 9-mer oligonucleotide, and the melting temperature was also adjusted as well. And this new adjustment will lead to the figure C right over here, where you can see the clear distinguishment between the 4 different coronavirus variants. So the sensitivity and the specificity of the RT-qPCR assay was also determined. The specificity was shown on the left figure with the A's and B's. The A will show the application curves of the respiratory viruses. 4 of them are the human coronaviruses mentioned before, as well as 9 different respiratory viruses as well, including influenza A, influenza B, and etc. As for the figure B, it's also a photo of the melting curves for the respiratory viruses as well, showing also the 4 human coronavirus variants as well as the other 9 respiratory viruses. And you can see that there are positive curves for the 4 human coronaviruses, but none for the 9 respiratory viruses. And on the right side, we have the sensitivity for the 4 coronavirus variants as well. And as you can see, there are very clear distinguishments and clear results for both the sensitivity and the specificity, determining that the RT-qPCR is really an effective way for detecting the human coronaviruses. So compared to the other research performed for human coronaviruses variants, this more new research was performed under the title of a new coronavirus associated with a human respiratory disease in China. And this research uses the RT-qPCR to determine the presence of SARS-CoV-2 in a single patient. The patient himself was admitted to the hospital on December 26, 2019, roughly around the start of the new infection. Due to the severity and the abnormal conditions of the infection and symptoms displayed by patient, the bronchoalveolar lavage fluid, or also known as BALF, was obtained from the infected patient for the RT-qPCR. And as you can see in the figure below, the genome sequencing for the SARS-CoV-2, are also known as WHCV in the figure, is depicted, as well as the other variants, one for the coronavirus in bats, and another variant called SARS-CoV-2. And you can see some similarities and differences between these three genomes. Involving the BALF sample from the patient, the RT-PCR was used to determine the presence of other respiratory pathogens or viruses such as influenza A or influenza B, either from the Victoria lineage or the Yamagata lineage, and etc. However, as you can see in the figures here, for A, B, and C, D, the results show no presence of the tested viruses or the other respiratory pathogens. Therefore, the BALF sample was tested with clinical samples that were at least positive for one of the following viruses: the influenza A, influenza B, the human adenovirus, and numerous other respiratory pathogens, and different coronavirus variants as well. And as you can see on the figure 8, that there is a positive amplification, and this amplification is going to be the standard plasmid of the human coronavirus, the SARS-CoV-2. And with this amplification curve, the DNA concentrations for each of the amplifications were determined, and a linear fitted curve involving the threshold cycle to the DNA concentration was determined. And seen on figure 3, the presence of the SARS-CoV-2 in the BALF sample was observed when the sample was produced from the positive amplification, while the positive control of the SARS-CoV DNA standard was used. Although the RT-PCR produces a high chance of detection, many other tests, such as the antigen and antibody tests, are used, especially due to the cost efficiency and the simplicity of the test itself. Another disadvantage for the RT-PCR is the fact that there are possibilities of false negatives or false positive results, although it is highly unlikely. And as the coronavirus has multiple different variants and strains, the mutations of the primer and the probe regions within the SARS-CoV-2 genome sequencing can actually result to errors as well. And in addition, the lessened sensitivity of the SARS-CoV-2 during the early infection stage can also result to false readings as well, which gives the RT-PCR great disadvantage. However, this could also mean that there's a lot of improvements that can be made to this testing method as well. And throughout the few years, there has been an increase of research performed for the new variants of the coronavirus and the different human coronaviruses. And based on the recent research conducted for the coronaviruses using RT-PCR, there are a lot of benefits of using this particular testing method, as mentioned before, the different sensitivity and then the specificity as well. So the RT-PCR has been shown to be promising testing method, and it is still currently the most commonly used testing method to detect SARS-CoV-2 and other human coronaviruses as well.